Right. Well, hello everyone. Thank you very much for coming, and hello also to the Medical School Hamburg, whom we see on the screen here. So today is our Trinity Term Brain and Mind event on science in the brain, after we've done reading in the brain and maths in the brain so far. This will be a hybrid event, as you can see. In the first half, we will have talks in neuroscience and psychology broadcast to us from Hamburg, where, as we can see here, Dr. Anita Avramidis will be hosting this event. And in the second half, after a cup of tea and some cakes, which you will find outside, Professor Simon Saunders will speak to us about science and philosophy from Oxford, and we will have a Q&A where we can ask both Professor Saunders and our colleagues in Hamburg questions and converse with the Medical School Hamburg. So thank you very much for coming, and I will now hand over to Professor Avramidis.
Hello, thank you, Barnaby. Can you hear me well?
Yes.
Good, and you can see me. I'm going to stare at that camera and hope you can see me in Oxford. Barnaby didn't tell you that he is actually from Hamburg, which is very odd because he's in Oxford and I'm in Hamburg. I live in Oxford. But once again, I'm overlooking the Elbe Delta, and it is an absolutely gorgeous day, and the sun is shining here in Hamburg, and I hope it is in Oxford as well. As Barnaby just explained, for the past 3 years, Brain and Mind has been run in collaboration with a medical school here in Hamburg. And we tend to do the spring event hosted from Hamburg. Before I begin my introductory remarks tonight, I want to thank the Medical School Hamburg for their continued support and generous support for these events. And I want to thank the principal and the governing body of St. Hilda's for their continued support. And a big thank you to the IT people, both here in Hamburg and in St. Hilda's. And although we've had glitches in the past, I'm told everything's going to go very smoothly tonight. So here's hoping. Now, collaboration is what this event is all about. As those of you who are regular attenders will know, these events are about an intellectual collaboration between neuroscience, psychology, and philosophy. For some, bringing these disciplines together will seem obvious. To others, it's going to be a new idea. Perhaps it's a new idea for some of the A-level students who attend, and if I may extend a warm welcome to any A-level students we have with us tonight in Oxford. Now we hold one Brain and Mind event in each of the Oxford terms, that is in the autumn, in the winter, and in the spring. And we try to organize the events around a theme. This year, the theme has been education and learning. And in this connection, we had in the first term, reading in the brain, in the second term, maths in the brain, and tonight the theme is science and the brain. The Western intellectual tradition tends to trace its roots back to ancient Greece. While the work of someone like Aristotle is mainly studied in philosophy today, the distinction between philosophy and science in those times didn't exist. Aristotle is as much an early scientist as he is philosopher. Indeed, this was very much the case for many great thinkers until around the 16th century, when we begin to see a bifurcation between thinkers that are now thought of as philosophers and those that we think of as mainly scientists. Indeed, in the 16th and 17th centuries, that period is now thought of as the age of the scientific revolution. And it's a real question how we should think of the relationship between science and philosophy today. And when I was reading this through, I realized I'd left out psychology. But actually, psychology is a very new science. And that also has its roots in philosophy. I say it goes back to the 18th century, but I think if you look at psychology texts, they probably cite the mid-19th century. Yeah, but I think David Hume was an early psychologist, or at least is a thinker that is associated. Now, the brain sciences offers us a particularly sharp focus for considering the relationship between science and philosophy. One question we might consider is the following. While science tells us how the physical stuff of our brain works, there's a real question about how this relates to how we as human beings, whole persons, experience the world. How do we marry what science tells us with that experience? Think about this carefully. If you see a neuron firing in the circuit of the brain, there's nothing in that alone to tell you about the wealth of experience that human beings have. Pain, anger, love, belief. So how does science tell us about how we experience the world? And again, here's where psychology can come in, because it can be the interface between the two with its exploration of human behavior. I'll let the psychologists tell us about that. Now, while this may not be the question that all our speakers tonight will be addressing, at least directly, I think it's a question worth pondering as we listen to our three talks tonight. Now, our first talk is going to be from the perspective of neuroscience, and our speaker is Professor Maike Glitsch. As many of you will know, Professor Glitsch is, along with me, a founding member, a founder of this series. 10 years ago, actually, we came up with the idea for this. When we conceived the idea for this seminar, Professor Glitsch was a tutorial fellow in Oxford. She's now professor in the Department of Physiology at the Medical School in Hamburg, and is the reason why we have this collaboration. She's going to talk to us tonight about what science can tell us about how the brain works. Over to you, Maike.
Thank you very much, Anita. Okay. Whenever we want to understand how something works, we need to know what the thing that we want to study actually does. There's no point if you study the eye to play loud music to it, it will not react. So the first thing we need to understand is, if we're looking from a scientific point of view, what does actually the brain do? It's not moving forward. And the simple answer is, it tries to keep us alive. And really what it does is, it's a stimulus response thing. And that might not be very flattering to us, because that's what single cell organisms do as well. They get stimulated and they try to respond to it in a manner that hopefully encourages their survival. Obviously, we like to think we have more complex responses to that, and I'll look into that. So what do? We initiate responses to external stimuli, like avoid or seek nutrition, avoid poisonous foods. We also have internal responses or internal stimuli, organ function, for instance, increases or decreases in blood pressure, and our body, our brain, will try to sort that issue out. And then we have emotions. Emotions are very important because they allow us to form memories, and then we can avoid in the future, or we can seek out situations in the future. Another important function to stay alive is to be able to form memories and to store them somewhere, because it allows us to identify situations and to respond appropriately. We have semantic and episodic memories. Those are memories where we can retell things that happened in the past, and we can also remember facts. That's also important to tell other people, and thus allow survival of the species. But also motor and emotional memories, so we know how to do things, which will encourage our survival, to walk so we can run away. But also emotional memories, that didn't feel good last time, I'm not going to do that again. Finally, a consciousness, so we're aware of things that are happening to us, and that it's us that it's happening to. And then obviously cognition for survival, important are to be able to invent things, language for communication, and also social norms, so establishment of rules and then adherence to rules. And then finally reproduction, which is regulated by the brain. You wouldn't think so, but it is. That's not necessarily our personal survival, that is survival of the species. If we look at the brain, and I'm now looking at stimulus response, you can see very nicely that we have sensory input to the brain, which will be analyzed by the central sensory system. And then we have a limbic system that controls our body homeostasis, our emotions, and learning and memory formation. Our reward system tells us whether what we just experienced was nice or not. They talk to each other, they communicate with the decision-making system that will then initiate a response that can be either initiated by the limbic system or by the decision-making system itself. And our output that we have is either release of hormones or contraction of a muscle. This scheme that I have here is a functional model, so there's a lot of overlap anatomically. So one part of the brain might be involved in several of these functions that I've described here. So how do we investigate to understand? So what do we investigate to understand how the brain works? And there's one, what is the function of different parts of the brain? And you have here an MRI that shows the different structures of the brain. Does a different structure necessarily imply different function? It's one of the questions we ask. Does it matter where a junction is located within the brain? And also different imaging methods, so different ways of showing pictures of the brain will emphasize different aspects of the brain. So we need to be aware of that when we study the brain. We also want to understand how neurons work, which are the building blocks of the brain. So does structure relate to function? We have here on the right a picture of a cerebellum. That's this part here in the MRI scan. And you can see that different neurons can look very different. You have a Purkinje cell, which is huge, and then you have the Greiner cell layer, and those are tiny neurons that you can find there. So what do all neurons have in common, and where do they differ, and how do they differ? If we're now looking at studying brain function, we can first look at the cellular level. So how would we study individual neurons? We could, for instance, have cultures. So we can take neurons out of a context of the brain, and we grow them in the lab in a dish. That would be a primary cell line. We can immortalize them, or we can take transformed cells that have become cancerous and they can survive outside of the brain. Those would be then cell lines. We can do co-cultures, so we can culture different types of neurons or different types of brain cells. There's other cells in the brain than neurons. And then we can study the function. These cultures would be in 2D or in 3D, so we can actually look at how does the structure of the brain impact neuronal function. We can look at brain slices. We can take functional tissue, we cut it into slices, and then we can study connectivity very nicely between two neurons. We could do that in cultures as well to some extent. That would normally not be done in human tissue. We would obviously use animal tissue for that. And finally, more recently, people have started to develop brain organoids, where you take very often stem cells, induced stem cells from humans, and you try to grow a brain that way, or a mini brain that way. You can see some progress has been made in that respect. There are some issues with these models, but it's a good appropriation. It's the best 3D model that we currently have. So what kind of techniques could we use? And the first one is electrophysiology. The brain functions in electricity. So electrophysiology allows us to measure the electric activity of an individual neuron. In fact, we could measure the activity of one single molecule, an ion channel. And we can then study the impact that certain drugs or toxins might have on the activity, on the electrical properties of that neuron. We can also look at connected neurons. We can induce an action potential, a stimulus in one neuron, and then record what that does in a neighboring neuron to find out whether these two neurons are synaptically connected. And if they are, we can again put on toxins and see what that does to the connectivity. Obviously, we can also try to genetically modify these cells. I'll talk more about that later. And then we can do imaging. So we can look at pictures. We can try to identify proteins that are, where are they expressed in a neuron. We can do that at a level of a slice, that's the picture on the left, or we can do that at an individual level, even as low down as a part of a neuron. We're seeing a dendrite on the right there. So we can see what happens, we can trace proteins, we can do all sorts of things here. Electrophysiology allows us to look at the activity either of all currents, of all electricity across the neuron, or just across a single ion channel. I'm not going to go too much over this picture, but we can get data that look like this. This is an individual ion channel to date. Electrophysiology, the patch clamp technique, is the only technique that allows us to monitor the activity of one single molecule. Or we can compare electric activity across a whole neuron, and then we can look at how different neurons compare to each other, whether certain drugs work on one neuron but not on another neuron, etc. So what do we know about how neurons work? We know that they form synapses. We know that we have a presynaptic cell that sends an action potential, and then we have a synapse between the pre and the postsynaptic cell, and the postsynaptic cell receives a neurotransmitter, converts it back into an electric signal. We now also know that a given neuron can receive anywhere between very few to hundreds of thousands of synapses. A given neuron may contact 1 to thousands of postsynaptic neurons itself, and the number of synapses is not static, so they can change in an activity or an age-dependent manner, and they can increase and decrease. We also know that synapses are not just formed between an axon and a dendrite, as was depicted in the picture above. We can also have synapses between the soma and the axon, axo-axonic synapses, and also between dendrites. So synapses are not no two synapses will ever be the same. We also know that neurons have different shapes, and the shape, of course, dictates the function, or the function dictates the shape, however you want to look at that. Then we come to synaptic transmission itself. So we're now looking just at one synapse, one out of potentially 200,000 on a given neuron. And the idea is that we have at the top the presynaptic terminal. From the presynaptic cell, an action potential will open ion channels, calcium will go in the cell, we will have release of neurotransmitter that is stored in vesicles in the presynaptic terminal, and the neurotransmitter is thus relieved into the synaptic cleft that separates pre- and postsynaptic cell. The neurotransmitter will bind to postsynaptic receptors. These tend to be ion channels, so when they open, ions cross the membrane. Whenever ions move, we get an electric current, and thereby we have converted an action potential, which is an electric signal, into another electric signal. So this is the picture that students learn usually. In fact, it's far more complicated than that. We know now that neurotransmitter release at the synapse can also bind, that's figure A now, can also bind to autoreceptors, so receptors on the presynaptic part, and it can influence whether or not we release neurotransmitter. We have heteroreceptors which will respond to neurotransmitter that is released at a different synapse. We have here an example of an axo-axonic synapse. But in fact, it could be any neighboring synapse that releases a neurotransmitter, and if it can diffuse to a neighboring synapse, it may well activate both postsynaptic receptors and presynaptic receptors. So what I'm trying to say is synapses can have a memory for activity, and they may take activity across other synapses into account, so no synapse is on its own. Also, very importantly, synapses, the postsynaptic cell itself can release transmitter onto the presynaptic, so the postsynaptic cell can talk back. So we have to say goodbye to the idea that information transfer is unidirectional from pre to post. In fact, post talks back to pre, and neighboring synapses will have their input as well. And there's no time, unfortunately. There's a huge complexity of neurotransmitters and neurotransmitter receptors and possible responses that can be elicited by neurotransmitters. Just to give you an example, glutamate is the most common neurotransmitter in the brain. 70% of synapses will release glutamate. We have eight kind of metabotropic receptors. We have 3 different kinds of ionotropic receptors, and they all come with different variants. So we have at least 30 different kinds of receptors that will respond to glutamate alone. So we can build up tremendous complexity. And then we shouldn't think that the activity of a synapse is static. It will change. It will change in an activity-dependent manner, and it can change over different time courses. The examples shown here show that if you employ a particular kind of stimulus, you might get an increase in the activity over the synapse. LTP stands for long-term potentiation here. So we have potentiated the response across the activity across the synapse, be that the neurotransmitter, that we release more neurotransmitter, or that the postsynaptic cell can respond better to the neurotransmitter that is being released. Both mechanisms are possible. We can also depress, depress synaptic activity, and I've shown you examples here for long-term. In fact, we can have short-term So we can have changes in the activity across a single synapse from microseconds to hours, days, potentially weeks, months, maybe years. But we don't know that. And then the shape of a synapse can differ. So we have for excitatory synapses, so those are synapses that will make it more likely that the postsynaptic cell will fire an action potential itself. The postsynaptic part of the synapse can look very differently. It can have different shapes. And the synapses tend to progress from the filopodium shape to the cup shape, depending on activity and experience. And this is a nice example here from a paper where they exposed, you can see here dendrites, those are the long lines. You can see the little dots, those are the spines that we're looking at. They quantified the types of different patterns of spines that they see, and then they treat the cells and they look what amphetamine in this case does to synapses. And you can see that the synapses change their shape in exposure to amphetamine. So synapses, even a single synapse, will never be the same. The moment time passes and you have a thought, some synapses will have been changed. Some forever, others just for a few microseconds. So what else do we know? We know that a neuron has dendrites, they receive information. We have the soma, then we have an axon hillock. This is where an action potential is initiated. We have an axon that sends on the information to the axon terminals, which then make contact with the postsynaptic cell. And some, but not all, axons may be wrapped around by a lipid layer called myelin. And we know that the axon diameter and the thickness of the myelin layer determine the speed with which action potentials can be propagated. At least that's what we thought until we found out. So action potentials, we said, can only be initiated at the axon hillock, and they can only go to the right on the screen to the synaptic terminals. It now turns out action potentials can also backpropagate. So actually you can initiate an action potential you can see here with the blue pipette on the right, you can see an action potential, and you can see that with a very short time delay, we can also measure an action potential in the dendrite. So action potentials, like synaptic transmission, are not unidirectional. And we also know, this is a recent paper, that actually the diameter of the dendrites critically shapes the speed of the action potential. So the more we look at neurons and synapses, the more we learn about them. So this was just single cell level, what we can do. And I'm heavily biased towards electrophysiology because that's what I've done. There's other ways to study them. So how about the whole brain? What can we do? If we want to understand the human brain, what we can do is correlate injured or altered brain structures with functional alterations in living people. So if you have survivors of traumatic brain injuries, for instance, and I'm sure most of the medical students and psychology students will be, yes, I see nodding faces, will be familiar with Phineas Gage. You can see here the rod that he holds so very proudly once protruded his head. It went through the front of his head. He had prefrontal injury. I don't know if you can see, one of his eyes is injured. He can't keep it open. And since he had this accident and he survived, we know that the prefrontal cortex is essential for decision-making, impulse control, and also personality to some extent. We can also have World War I, for instance, was a great source of information about how the brain works. Those people who survived traumatic brain injuries, you can see here shrapnel in the head of somebody, of a survivor. Very often the cerebellum was involved. It's in the back of your brain. We learned a great deal about different parts of the brain. What do they actually do from the defects that the people experienced afterwards? Can they still move their arms, limbs? Can they now have appropriate responses, etc.? Or, of course, post-mortem analysis of brains. You can see here a stroke in a brain slice that will tell you very clearly what the cause of death was, and also that might explain why a patient before they died had certain issues. We can also, in living brain, use imaging techniques such as MRI and CT. They provide largely structural information. You can see that MRI and CT, different structures light up differently. CT scan tends to give faster results. We can also do functional activity. We can look at metabolic activity, PET and fMRI. These are examples here. And areas that are particularly active will shine up in different, either in colors here in the fMRI or PET. In PET, we'll have a heat map showing aspects of the brain that are more or less active. There is a conflict when we're doing imaging techniques in a living whole brain about spatial and temporal resolution. So we'll be hard pushed to get very quick information. We could not look at the microsecond scale or low millisecond scale. We can look with fMRI maybe hundreds of milliseconds, and the spatial resolution is nowhere near as good as we would have when we look at individual neurons. We can also look at EEGs and MEGs, which is either the electric or magnetic activity in the brain. Limitation here is that we can only look at parts of the brain that are on the surface. We will get traces like you can see here on the right. But actually, we've used EEGs and MEGs very successfully in order to monitor brain activity in real time. What happens here is that electrical signals are averaged out over an electrode, over the site of an electrode here. And of course, an EEG would be a way to determine whether a person has, for instance, epilepsy or not, simply because that would not show up in an MRI or a CT or fMRI. MEG is another way to, usually it's used together with EEGs in order, it has a very good time resolution, 10 milliseconds or faster, so it's much faster than fMRI. And they can be used in conjunction to create maps of where is what located in the brain. And then, of course, we could do psychology. I did not dare to venture into that territory. The next speaker will be much better equipped to do that. And of course, we can do animal studies. And before I go into animal studies, I just want to show some pictures, some nice pictures of what we know so far. So we can correlate now. We have so-called sensory cortex and motor cortex. So we can correlate, for instance, if I talk, I'm talking a lot now, so this part of my brain will be very active now, moving my tongue around. If you're going to touch something cold with your lips, this part of your brain will light up and will tell you, I'm feeling something cold now. This is kind of textbooks, all medical and psychology students will be familiar with this. We can also look at taste perception. So we now know that we can map different tastes to different parts of the brain, and whilst there is some variability between different subjects, we have here 3 different brains looking at different parts of the insular cortex here. You can see that sweet stuff tends to be in this very similar, sweet taste is detected in a very similar brain area, and umami as well. We can also have follow-up where we hear tones, single tones. So this is now an auditory map. We call it a tonotopic map. Low tones are shown up in red here, high tones are shown up in blue, and you can see again that we can construct functional maps which part of the brain will be specifically responding to high tones and which will be specifically responding to low tones. This is the auditory cortex, the primary auditories that we're looking at here. So we have a very good idea about mapping. We can also look at other diseases where sometimes people are born and parts of the brain are missing. There's a very intriguing disease or condition called Urbach-Vitis syndrome, where you can see on the left here a healthy or a not affected individual, and on the right an individual affected by Urbach-Vitis syndrome. And what they're missing is the amygdala. And these people who do not have an amygdala are just never scared. You can't terrify them. They will not respond to scary situations. They can subject them to having spiders crawl across them and they will just find that really hilarious. They will not be terrified. You can make them watch horror films and they will not be scared. So we now know amygdala is essential for emotional regulation, in part based on these patients. And we also know, again, from injuries or from stroke studies that we have different parts for language production and language comprehension. Wernicke's area is essential for language comprehension and many different things. This is somewhat a simplification. And so this is here the place where people understand when you talk to them and can produce sentences that are understandable to us, so to non-affected people. And Broca's area is the language production. So you understand everything, but you cannot produce language. You can also not produce sign language, just in case you're curious. And you cannot write things down. So you've completely lost the ability to communicate in very bad cases. So these are things that we could elucidate with just the techniques I've shown you. The big question is now, of course, can we use animals to study brain function? And the biggest question you're going to ask is, well, which animal do you choose? There are many things to consider. The three biggest ones are ethical, scientific, and financial considerations, not necessarily in that order. Most commonly these days, people will use mice for brain function studies. Before that, it used to be rats. Rats are considered to be closer to humans than mice. Rats are bigger. They're actually much easier to study, but they're not easily genetically modified. So that kind of saved millions of billions of rat lives, but it cost mice dearly. So we now use mice, and of course they're mammalians, so that's great. And what can we do? What do I mean by genetic modification? A genetic modification would be when we mutate a gene, which results in a mutated protein, and then we see what that does to the animal. It's nice to trace brain development and aging, or we can look how a cell or a tissue or an organ or a whole animal function is affected by a mutation. A mutation itself could be an altered function of a protein. It may work better, it may work worse. We may completely lose its function, or it may gain a new function that it didn't have before. We could just label a protein so that we can see where actually is it expressed, either in the whole brain or in a brain area or within a cell. And we can alter the extent of a mutation. We could simply modify ultimately just one single amino acid, or we could introduce a stop codon, which means that we're making a smaller protein than it would normally be, or we can take out a complete functional domain of a protein. Proteins are made up of different functional domains. One domain might allow to bind to another protein, another domain might have an enzymatic activity, yet another domain might be there to regulate its activity. And so we can take out one of these functional domains and see what that does. We could also, in a mouse, introduce the same mutation that we find very commonly to be happening in human disease. So we can try to replicate human disease in a mouse. The question there is, how do we know that the protein we're mutating has exactly the same function as it does in a human, and we don't? Yeah, okay. We can also remove genes. We can introduce new genes. With optogenetics, we can light activate neurons in living animals, so we can influence behavior by shining light on a mouse. We can replace. We can do a lot of things. We can do it either that all cells are affected from birth, or we can do it that only one particular cell type is affected by it, and only at a particular time in development, and only for a certain period of time. So we can be extremely specific nowadays when we alter neuronal behavior. In recognition of the contribution of mice, there's a monument to mice in Russia. You can see it's knitting a DNA there. Mice have an advantage that they're used because they're cheap to keep large numbers of offspring, short generational span. We can also look at other animals. We're looking at zebrafish. They're translucent, so you can see very nicely where the neurons are. We can use Drosophila because we can see proteins again. They have a very limited number of cells, and we can even use worms to study brain function. Very rarely primates for obvious reasons. The problem is we don't know what the different size of a brain does and what different structure of the brain does that we have between mice and humans. So for instance, take the example of an elephant brain. An elephant brain is 3 times the size of a human brain. The cerebellum, that's the big part here on the left, on the right, contains in elephants 79% of all neurons, in humans only 50% of all neurons. The cortical part, which is for processing, contains 3 times fewer cells in the elephant than in humans. So size isn't everything. It seems to be complexity that matters. And here we have complexities usually taken at what does the cortex look like. But if you look here, you will see that marine animals seem to have then more complex brains. So they seem to be smarter than we are. Next question is, can mood disorders or psychiatric conditions actually be accurately studied in animals? And how about neurodivergence? We once had students telling us, oh, we've got a mouse model for autism. The mouse never looks and interacts with other mice. And then another student comes, oh, we have a model for depression. We have a mouse that never interacts with other mice. And the question is, okay, does that work? And then how do ages compare? We're considering mice adult after 3 weeks. That's when they reach sexual maturity. But how does it compare to humans? Obviously, there's problems. The methodology that we use to study the brain or neuron limits what we can learn. The more we look, the more complex everything gets. Two neurons are never the same in terms of location, protein expression, connectivity, and experience. Never, ever. Same is true for synapses already. Neurons are not static. They change with time and or in an activity-dependent manner. And brain areas can have many different functions, and a given neuron can take part in a number of different processes. And this is the last slide. What we still don't know, this is just what I would like to know. Why do we sleep and dream? We don't know. How are memories stored? We don't know. How do glial cells shape brain function? That's 50% of all cells in the brain. We don't know. I would really like to know that one. How does biological sex affect brain function? So is it the different chemicals, the sex hormones? Is it different mechanical environments? Male brains are softer than female brains. Is it we have different presentation of neurodiversity? Why? Is it social? Is it not? Is it biology? And different likelihood of having certain brain pathologies. Alzheimer's far more often in women, in biological females, and Parkinson's far more higher likelihood in biological males. And this is it. Thank you. So a lot still to learn.
Excellent. Yeah. I hope everything is smooth over there. I'm not hearing anything from Barnaby, so I'm assuming it's nicely transmitting. I'm not going to take any questions. We're going to go straight on to our second talk this evening. It's going to be from the perspective of psychology, and our speaker tonight is Professor Andrew Tolmie from the University College in London. Professor Tolmie is Chair of Psychology and Human Development at UCL Department of Education and Deputy Director of the Institute of London Centre for Educational Neuroscience. You'll see from the programme some of the interesting work he's done in connection with the teaching of science. And we're delighted that he's come over from London to be with us this evening in Hamburg and transmitting back to Oxford. Professor Tolmie.
We promise no technical glitches, but it's the humans that mess things up generally. So what I want to do this evening is to kind of really flip over from what Maike has just been talking about. So that I think can be seen as what science tells us about the brain. I'm going to be talking really about what the brain tells us about how we understand science. And in particular, the rather surprising role, perhaps, of imagination in understanding science. So that's going to be the focus for this evening. And I'm going to be talking particularly about primary age children. Let me just start though by showing you this video. Can you click on that to run it? Thank you. So I'd like you to just watch what's going on here and think about what's actually happening. I'm not going to ask you what's happening. You probably recognise that we've got two different types of paper being dipped into water here. One is tissue paper, one is blotting paper. They are both taking up water. They're doing so at different rates. You would recognise that as being an instance of what we call absorption, I'm sure. But what I want you to hang on to here is how you understand the nature of the difference between the reaction of the two materials to water. Why is one faster than the other? How do you understand that? What are you actually thinking about when you're trying to understand what's going on there? So just trying to hold on to that kind of notion as I go through the other parts of this talk. So one important place to start here is really why we're interested in how children learn science. And basically this is because of the importance of scientific understanding, not just for jobs in the science economy, obviously it's an important preparation for that, but also in terms of what we call scientific citizenship, so the notion of people becoming adults who understand something about scientific issues that can respond appropriately to those in terms of engaging with the public decision making, so things like climate change, vaccination programs, and so on and so forth. So obviously we want to start preparing children for learning this kind of understanding, gaining this kind of understanding as early as we can, in primary school in particular. It's not so very straightforward in terms of how we engage with that educationally, because science is a complex activity. It involves understanding things about how you actually conduct the kind of research that leads to scientific conclusions. There's a lot of technical stuff there. There is also importance of understanding kind of scientific knowledge. So, the Earth goes around the sun and things like that. So there's a kind of knowledge element. But there's also a kind of core focus, really, of science on understanding causes, what leads from one thing to another, what is the kind of connection between one event and another event. And it's that kind of core focus on causality that we have been thinking about very much in terms of children's understanding of science. And indeed, we've shown, just to kind of give a kind of glimpse of that, we've shown in our own work that actually, even if you take kind of broad measures like are used in the TIM study, the Trends in International Mathematics and Science Understanding, if you look at those kind of general measures of science and compare them to the measures of causal understanding that we've used in our research, and particularly things to do with explanation, understanding of mechanisms, those are well correlated with the kind of general understanding of science. So causality is a kind of big focus for understanding in primary age children. Now, in actual fact, when we're thinking about what's happening in primary school, this is something that has been built on from much earlier in development. We know that from ages 3 and probably younger than that, children are very good at understanding connections between events. So they recognise that a switch will turn on a light, for instance, that one thing is actually kind of associated with another event, that kind of causal connection. And we almost certainly share that understanding with many, many other species as well, because it is evolutionary advantage. What we want to do when we're getting into primary school, though, is to expand that understanding of connections between things into an understanding of the mechanisms that lead from cause to effect. What is it that's actually going on at the kind of level of process that's leading from one thing to another? And the importance of that is that that's really starting to build up a more kind of theoretical understanding that can be then provide the basis for formal science in secondary school. And this is particularly the case when we're looking at the kinds of natural processes that are involved in science typically, where we're talking about extended causal processes. Things don't just happen instantaneously, they happen over a period of time. So for instance, the absorption example I was showing at the start, or things like water evaporating, salt dissolving in water. These are all things that happen over a period of time. If you think about the Earth going around the sun, it's a process that's been going on for a very, very long period of time and will continue to do so. So most causal processes are actually extended over time. So why does that matter? Well, the point about that is that actually the kind of processes that are actually operating at a causal level are not things which are typically observable, a point that the great developmental psychologist Jean Piaget noted in his 1974 text. So when we're talking about these kinds of processes, things that happen over time, there are observable dimensions to that. So you think about a child throwing a ball to a parent. There are things like, who's doing the throwing, what objects, what movement is taking place. But if we want to understand what's actually happening at a causal level, we have to engage in a different way with kind of unobservables. So things like the force of the throw, the air resistance, the operation of gravity. These are all the things which are actually shaping the actual process of the ball being thrown and caught. So how do children come to understand that kind of unobservable mechanism? And for a long time in education, I think there's been a strong tendency to think that the best way to foster that kind of understanding is through verbal instruction. You simply tell children what's going on here and they will understand it. Well, actually, maybe not. I think there's a growing body of research that says that telling children things like this is actually kind of unhelpful. At the best, it can lead to a kind of unprocessed understanding where you don't really know what it is you know. But at worst, it can actually lead to misconceptions that actually get in the way of proper understanding of what's actually going on. And in actual fact, as I come on to explain a little bit in the rest of the talk, evidence really suggests accurate grasp of mechanism stems instead from non-verbal processes. So the role of imagination that I was alluding to at the start. Let me give you an illustration of that. What have I done? I pressed the wrong thing. Okay. There we go. Okay, so I'm sure you all know that weight makes no difference to the rate at which objects fall. A lot of adults tend not to believe that, actually. Here's a nice study from a colleague of mine, Christine Howe, and her PhD student, Michael Haast, from a few years ago, looking at children's 5 to 11-year-olds' ability to either explicitly predict or simply kind of register recognize the relative rate of fall of heavy and light objects. So if you ask children to predict whether a ball, heavy or light ball, is going to be dropping faster or at the same speed through a tube, as is on the left-hand side here, it's a kind of verbal response, kind of conceptually driven response. So they've got to choose whether A is going to fall faster, B is faster, or they fall at the same rate. That's the kind of conceptual response, verbal response. And then on the right-hand side, they simply see videos of objects falling, and some of them are kind of manipulated so that the heavier object falls faster, some of them are manipulated so the lighter object falls faster, or they are falling as they would naturally at the same speed, and they're just asked simply to say, does it look right? Which one looks right? And what we find from this is that as children get older through the primary school age range, as far as the conceptual response is concerned, they actually get increasingly worse with age. So they start off actually being kind of reasonably happy to believe to say that things will fall at the same speed. But as they get older, they typically say the heavier things will fall faster. And we think that's because they've actually heard this from a lot of people. People perpetuate this knowledge that heavier things fall faster. And so they just simply kind of will go with that. The recognition response, on the other hand, stays accurate. It doesn't change with age. And so we get this kind of weird situation with older primary age children, 10 and 11-year-olds, where what they say is in direct contradiction to what they actually will recognise as being the right event. So the verbal stuff is getting it wrong, because it can be misleading, whereas the perceptual stuff that's not relying on any kind of verbal processing seems to stay accurate. Okay, so why is that? Delving a little bit into Maike's area for a moment, thinking about this during, in terms of the kind of neural activity that's involved here. What we find is that if you look at the activations that occur during non-verbal tasks, these are actually occurring in different areas of the prefrontal cortex and areas relating to sensory encoding in the posterior cortex. So they're recruiting different areas of the brain when you're responding to non-verbal tasks. And so a kind of instance of that is the idea that we have different streams of activity within the brain for processing where and what information, what we call the dorsal and ventral streams. So when you're kind of thinking about what something is, which is something that is usually kind of verbally labelable and processed in that kind of way, that's happening in one area of the brain. The kind of more dynamic information, where things are and where they might be moving over time, that's happening in a different part of the brain. And in actual fact, it's worse than that, because that kind of dynamic information we know is actually rather harder to capture in everyday language than the kind of static information relating to object features. And because we're talking about change over time, when we're talking about causal processes, it's therefore that dynamic information that we're reliant on, but we find it very difficult to capture in everyday language. So we have to deal with that in a different kind of way by imagining, if you like, what's going on. So what we've been working on over the last number of years on relating to this is that the key elements of understanding science are, first of all, careful observation. So you're actually picking up the visual or other type of modality, sensory modality of information. So you're carefully observing what's going on, tracking that information, paying attention to changes over time and what it is that's actually happening at that kind of level. So what we call spatial temporal cognition, and then construction from that information of imagined processes that are actually dynamically underlying the observable. So this is the kind of unobservable bit. You have to imagine what's going on. You can't actually see it. You have to actually kind of think about it in that kind of imaginative sense. Okay. So that's the kind of general idea we're talking about verbal and non-verbal cognition. And we've shown in our research that actually there is this kind of switchover between what is important in understanding causal processes. To begin with, it does seem to be very kind of language driven. So you look at the kind of lower performances in terms of causal understanding, those are quite strongly predicted by verbal ability. But as you get up to the kind of higher levels of understanding of causality, the kind of better understanding of mechanisms, it's actually non-verbal processes, non-verbal cognition that's taking over as the key predictor of what's of that understanding. And to illustrate, sorry, it is me. I think I pressed the wrong button. Thank you. Another way of capturing that is in this graph here, where we've got the line at the top showing increasing levels of causal understanding, of better understanding higher up. And as you can see from the lines underneath, which show the degree of correlation between verbal cognition, verbal ability and understanding of science, that's the blue line, and non-verbal cognition and understanding of science, you can see that as performance gets better, they swap over in terms of what's actually driving performance. So the non-verbal starts to become more important to understanding around the kind of midpoint from the scales that we're using. And when you're getting up to the top levels of performance, it's really non-verbal cognition which is driving understanding. So again, back to that same kind of idea of dynamic imagination of what's going on. The other thing to pick out from this slide is that actually we find this is associated with levels of parental education as well. There is something, we don't quite know the story on this yet, we're doing work on it just at the moment. We don't quite know what it is, but there's something that better educated parents are doing that's promoting better non-verbal cognition, and that's actually something which seems to be important in terms of science understanding. So it's not to say that language isn't important, but it's really just important in terms of the acquisition of more specialized scientific vocabulary later on. And that only comes to the fore once those kind of non-verbal representations, those imaginative perspectives have actually become more established. And without going into the technical bit of the kind of this table at the bottom, what it's showing is that basically if you look at prediction of causal understanding from everyday language, from scientific vocabulary, these seem to be independent, but when you put in non-verbal cognition, the impact of scientific vocabulary just disappears, suggesting that actually all the scientific vocabulary is doing is allowing the non-verbal representations to be accurately captured, and that's the kind of key point of its function, if you like. So it's the non-verbal that's really, sorry, it's because I'm not looking at my thumb when I'm pressing it. And in particular, what we've also found in more recent work is that it's temporal attention that is especially fundamental here. So this is a study in which we showed a moving helicopter moving behind an occlusion and then appearing again the other side. sometimes at the right time in the right position, sometimes at the right time but the wrong position, sometimes at the right position but the wrong time, and sometimes everything being wrong. And what we found was that that actually relatively few people were good at spotting the temporal disruptions. Only about 10% were accurate at doing that. But those 10% were the ones who performed better on causal understanding. So it suggests that temporal attention, watching those processes carefully, paying attention to what's actually going on and building up a representation based on that observation seems to be the kind of critical thing in terms of understanding of science. So what that suggests is we need, if we're going to think about this in terms of primary education, we need a different approach to that which has been typical in primary science teaching, one that's based on encouraging imagination about hidden processes, based on close observation of what's going on, and linking results, resulting ideas to scientific vocabulary. So as an illustration of that, something piece of work that I did with one of my former PhD students, is to actually try that out, a lesson within primary school where what we did was we showed children, whole class basis, a stone and a tomato being dropped into a tank of water. The stone sinks straight to the bottom, the tomato goes down nearly to the bottom, if you get the right tomato, and then bobs up to the top and floats. What's going on? And we get them to describe what they've seen, to make sure they've observed all the kind of key dimensions of that accurately. We then asked them to play out a game which encourages imagination about what's going on. So we got them to threesomes, where one is the stone, one is the tomato, and the third is the water. Talk to each other. What's going on here? Where is there a difference here? And then we provided them with a new set of objects to make predictions about what was going to sink, what was going to float, and why. And we provided them with a diagram shown at the bottom with some vocabulary to start attaching those ideas to. We did this work with 180 children in years 1, 2 and 3, the first 3 years of primary school, in a very deprived area of Oxford, in fact, called Blackbird Leys, that some of you may be familiar with. And within half an hour session, we had year 1 children talking about water pushing the tomato up. Kind of, really starting to get into the mechanisms that are actually involved. At the end of that lesson, one of the teachers said to us, "I had no idea these children could say this kind of thing." So just simply by encouraging the right kinds of things, we can perhaps hope to promote better understanding of science in a really kind of more fundamental way from early on, and in children coming from disadvantaged backgrounds who are less likely to be able to engage with this in appropriate fashion to begin with. So making imagination central seems to be the kind of key part of this. And having started all that work a little later, I came across this quote from Albert Einstein, which actually said it all. Imagination is more important than knowledge. Knowledge is limited to all we know and understand, while imagination embraces the entire world and all there ever will be to know and understand. So Einstein understood that imagination is central to doing good science. Thank you.
Wow, fantastic. Whoa, is that okay? Is that okay? Thank you so much. Are we having a bit of feedback?
No.
Is it all
good? We hear you very well.
Excellent. Sorry, we're getting a little feedback here in Hamburg. Can we just clear that up? What I want to say, perfect example. We don't ask our speakers to coordinate their talks. They don't know what they're going to say. I just want you to hear how different subjects approach kind of topic. Now, this I've been doing it. We've done things like perception, we've done belief, we've done pain, done fiction, all sorts of topics like that, where you can see there's a topic that each subject is giving its perspective on, how it studies it. This time we're just looking at science, and it's a little more free how we're going to approach it. And yet, I think these two talks went together just beautifully, and I'm sure the third one will equally. So I'm now going to take questions. Feel free to ask either of our speakers. I'm going to start in Hamburg, then I'm going to go over and ask Barnaby in Oxford if he can get some questions from that audience. So let's start here if you have a question. And we have a microphone. You can use this one, I guess. Do we have a question? Okay, if people are being shy here, keep thinking, and I'll go over. Oh, we do have one. Hold on, Barnaby. Okay.
I have a question for you, Maike, actually. I was just surprised by one thing you said, which was that we don't know how memories are stored. Now, I mean, my area of research is working memory, so I'm looking more at attention. So that's that. But things like episodic memories, I thought we understood quite well how they were stored in the neocortex in terms of networks.
Not the mechanism. So I'm not sure. So we think it's probably synaptic activity and patterns, but nobody's ever proven it. Not to my knowledge.
No, that's really interesting. I mean, I'm not an expert in memory, so I don't know anything you don't know.
So we talk about the engram, and then how does it move from the hippocampus to the cortex, and how does it know where it needs to go, whether it was a visual memory or an auditory memory? How does it know how to get there?
So how does it organize?
The whole thing.
Okay.
And are proteins being made? Is it really patterns of synaptic activity? We still actually, I've not come across any paper that says it's definitely that. We have neurons that we call the Jennifer Aniston neuron, right?
Oh.
The neuron that anything to do with Jennifer Aniston, that neuron supposedly will be active. So whether you hear the title music from Friends, or you see a picture of her, or you hear her voice, or you see the haircut, that neuron should be active. But then how is that information stored in that neuron? How is it proteins? What is it about that? Is it the synapses at risk? We don't know, especially since synapses are so whimsical and they keep changing all the time. But actually we don't know. Can synapses be made permanent? We don't know.
That's really interesting.
There's a lot of things we still don't know, to my knowledge.
Thank you. Barnaby, do you have any questions there?
Yes. That was loud. Sorry. We have a question from Professor Saunders.
Right. Thank you very much. Fascinating talks. I have so many questions, but could I just ask a quick question each? So just on this issue of the Alliston neuron, you don't mean singular. You surely meant that there were various clusters of neurons distributed over the brain. It's not the case that you had really somehow tied it down to single neurons. So, I mean, that's a question for you. The other question I have is this remarkable, really opened my eyes to a whole dimension of developmental psychology, I suppose, with science learning. It's been absolutely a very fascinating talk. But I was struck by the specific thing you're working on right now, and this business of parents who are better educated, being better at facilitating the more causal competences or non-verbal competences. I just wondered, can you sort the parents in terms of their level of education also by arts and sciences?
Sorry.
And specifically actually humanities.
No, it doesn't seem to be a matter of what kind of education people have had. It seems to be a matter of the extent of the education that seems to be predictive. We don't quite know really what it is that better educated parents might be doing. We suspect that it's something to do with providing, having the ability to provide more interesting visual environments, more stimulating environments that might lead then to kind of more careful attention to what's going on, and that leading in turn to better developed non-verbal cognition. We're trying to do some work just at this very moment in time. Data collection is just about completed, where we're trying to look at that in rather more detail, but I don't have any more specific things to say about that, but it doesn't seem to be the area of education, it seems to be the extent that seems to be the interesting thing.
Okay, coming back to the Jennifer Aniston neuron, I completely agree with you. It has to be a population, but it is referred to as the neuron. And if it's a population of neurons, then presumably some would respond to haircut, others to music, others to her looks. But the talk is about the Jennifer Aniston neuron in that literature. I'm with you. I think it can't be just one neuron.
I mean, just to comment on that, I'm sure you're familiar with Jerry Fodor, a very famous philosopher of mind, especially in the 90s, I think of him and so forth, but very much pushing a view that does seem slightly, eccentric, that there are individual neurons in order for, and I think it was objects or proper names to have meanings. So it's just interesting that there were what I thought to be slightly implausible views of how the mind works. And so if you do end up with just a single neuron, that only when all occurrences of Aniston it lights up, then that really would be extraordinary. But I shared that I thought the thought, yeah, it must be a population. It can't.
Yeah, no. I completely agree. Completely agree with that.
What if that one died?
And it raises the issue of how is it stored if it's just the one neuron.
Okay, Barnaby, do we have another question? Oh, we have one here. I'll take one more from each, and we'll keep them short so we can have our break.
I have a question for Professor Andrew Tolmie. So can we introduce this way of teaching, like prediction, testing, and explaining in an adult or teenage age? Or if it isn't started like in the childhood, now it's difficult or impossible. This is because of the sensible and critics period of childhood and so.
No, I mean, I think we don't think anymore that there are critical periods for learning. We do think that there are sensitive periods for learning. So there are age ranges at which certain things, certain types of understanding are perhaps more easily acquired. That certainly is true in terms of language acquisition and things like that. But it's not defined in terms of, absolute essential. As far as this kind of understanding is concerned, I mean, the trouble is that we have only really been tackling this properly, I think, from a kind of cognitive psychology perspective for a short period of time. So we don't know what is important that might be happening when. There is something about temporal attention, and we know that that ties in with other things that we talk about a lot in psychology, and especially in educational contexts, to do with executive function and basic control of cognition, which has both non-verbal and verbal dimensions to it. Attentional processes seem to be very important for that development. It does look as if what happens during the first 5 or so years of life might actually be particularly important for shaping subsequent development. We know, for instance, that social disadvantage impacts this kind of executive function. Children from more disadvantaged backgrounds tend to perform poorer, and that persists into secondary school. And actually, maybe in some respects, the gap widens during that kind of period of time. So it does seem as if the first 5 years maybe are very kind of important. As I've said before, in response to the previous question, we don't quite know what it is that might be happening within the home environment that is particularly important, other than kind of slightly a guess that visual stimulation, interesting environments might be particularly important. So that kind of address what you're asking.
Yeah.
Thank you.
Okay, just one more question quickly in Oxford, please, Barnaby.
Yeah, we have a question over there. Thank you.
Hello, this is a question for Professor Saunders.
Hold on, Professor Saunders hasn't spoken yet.
Sorry, I've got his name wrong, the man from the Institute of
Professor
Education.
Tolmie. Okay.
Sorry.
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 That's okay. I'm sorry too. Hi. I am a retired head teacher of a school for children on the autistic spectrum. And one of the things that interested me in what you said was about the use of imagination in teaching science. And I think, from my own experience with these particular children, some of whom are highly intelligent, that use of imagination in teaching can really work right across the curriculum. And I wonder what you think about that. I know you're in the early stages of this work, but it seems to me that some of the things that you say seem to be missing, like interaction perhaps with parents on a social level, and obviously children with autism have a social disadvantage because of their particular disability. Imagination does encourage socialization and interaction in any situation, but particularly in learning. And I wonder whether you feel that the work you're doing could be spread right across different parts of the curriculum.
Yeah, I mean, absolutely. I mean, I think the interesting thing from my point of view, I'll come to the specific connection to autism in just a moment. But the interesting thing from my point of view is, as somebody who's focused a lot on non-verbal cognition, is just how little attention has been paid to that as part of the educational processes generally. I mean, verbal cognition has been privileged educationally for a very long period of time, and there are obviously reasons why that's important and why that's happened. But it's illustrated perhaps best by the fact that we have actually a really clear kind of taxonomy for many aspects of verbal cognition. We don't even have a taxonomy really, particularly for thinking about non-verbal cognition. We don't know quite how to break it down, what elements of it are important. So I think there's a kind of an educational advantage to encouraging these non-verbal processes, non-verbal cognition, imagination as part of that. Across the board, it seems to be something that might actually be very fundamental, but hasn't really had very much attention paid to it previously. I think there are some particularly interesting things about people with autism in terms of their engagement with things like scientific processes, causal processes. The ability to focus on the kind of detail is actually exactly the kind of thing that is generally been seen as more a strength within autism. And also the kind of filtering out, the kind of paying less attention to the verbal side of things, is perhaps is a reason why certainly there has been some tendency, I think, for people with autism to do actually quite well in terms of scientific careers. So that's a speculation really, but I think certainly there is some kind of connection there that's interesting.
Thank you.
Okay, excellent. If you have more questions, there's going to be plenty of time at the end if we keep to our schedule. So I'm going to let you go and have tea, Barnaby, and I'll see you back at 6:30-ish. Okay?
Perfect. Thank you very much.
All right.
Right, well, please feel free to help yourselves to some tea and cakes outside and before you.
Okay, Barnaby, how are we doing?
We are doing fine.
Are people coming in?
People are all in, and we're almost ready to go. I'm just going to go shut this door, and then we can start.
Sit down. We're about to start again. Come sit down. Excellent. Hang on, Simon. I'm going to introduce you from here. Barnaby, tell me when you're ready.
We're ready, so if you want to go ahead, you can do introductions.
Okay. Can everybody be quiet here in Hamburg? Are we ready? Okay. All right. So we're back now, and we're going to have the final talk of the evening, which is going to be philosophy. And we're very pleased to be able to have with us Professor Simon Saunders. He is in Oxford. He is an emeritus professor of philosophy of physics at Oxford University and a fellow of Merton College. Now, I should explain that in Oxford, you cannot do philosophy on its own. You have to do philosophy with another subject. So we have philosophy in modern languages, philosophy in politics and economics, philosophy in linguistics and psychology, philosophy in mathematics, and we have philosophy of physics. I've only mentioned some of the subjects. Now, Simon is known for his work in quantum physics, and tonight he's going to give us a talk entitled Science as Perception, Experimental Data, Sense Data, Their Sameness and Difference. Over to you, Simon.
Thank you, Anita. And it's a pleasure to be able to talk to you. And I do find some synergies between what I'm going to say and the last talk in particular, so actually with the first as well, so that's very pleasing to me. So I have this topic. It's primarily a story of two points of view, if you like, two figures in philosophy, two dominant figures in philosophy of science over the last 50 or 60 years, although Bas and Frossen more dominant the last 40, and Van Quine perhaps the previous 50 or so. So these are major figures, and philosophy is often moves by fashion, and they may recur in different forms, I suspect, in decades to come. But they were both empiricists. So this was the fundamental tradition in philosophy of science in the 20th century. It began with the Vienna Circle, the logical empiricists, logical positivists, this whole movement of which Quine was an ardent follower and contributor, staying with Carnap during his time. I think it was in Prague when he was writing logical syntax of language, one of those high points in 20th century early philosophy of science, and proceeded to champion empiricism, but modified. So this empiricism that was so dominant, it combined two features. One of them was a focus on sensory stimulus, perception. All knowledge comes via the senses. Absolute dominant theme going back to the empiricists, people like Locke, Berkeley, and Hume. But the other theme was anti-realism of some sort, some measured sort, and it came and went. How measured was the anti-realism? It was fairly a sort of surrealism or an irrealism almost in the work of Carnap. Somehow floating above existential questions or existence questions, ontology, what really is there, or even what is truth. And Van would have none of that. He made the great rebellion against the logical empiricist movement in the late 40s and 50s. Well, in the eyes of many, mine anyway, was one of the major contributors to its collapse by the 60s. Now, Bas van Fraassen was very much there in his early years where the huge, with this collapse of empiricism, a new form of realism, scientific realism. And this was taking everybody's, blowing everything away before it somehow, very fast-moving and getting more and more pervasive and so forth. And Bas held out against that, and he wrote a book, a very important book, The Scientific Image, published in 1980, which really took a contrary stance and said, look, no, there is an alternative to this kind of scientific realism and what it involves. And that alternative is a form of empiricism. He calls his position constructive empiricism. But it is not empiricism in the traditional sense, because I have no interest at all in sensory stimulus. Or, well, curious where he did, because science had to tell him what was and what was not directly observable. But other than that, he had no particular epistemological interest in sensory stimulus and how we arrive on the basis of sensory stimulus at, for example, our grasp of ordinary objects. And that had always been part of traditional empiricism in philosophy, in modern philosophy. So quite an interesting divide between the two, because what Quine did, whilst retaining that focus that laser focus on sensory stimuli. He was a realist. So I hope I've set up an interesting apposition for you between these two people. Alas, they were separated in years by about 30 years. And I think it was just enough for them never to really have to, I want to say the word confront each other or have a serious debate with one another, as so often happens in philosophy. It's a shame, and I think it needs to be more that elderly great philosophers, as Quine was, when Bas van Fraassen was a young philosopher, can answer to a sort of, new generation mavericks or whatever, or critical and so on and so forth, because it would have been a fascinating debate. So, and I think the age gap was just sufficient such that, and Quine, it was perhaps, he didn't really write explicitly on Van Fraassen, so they had very little interaction, one or two pieces, that's all. So this is a story about something that should have happened and never did, and I hope will go on to happen. So here's the two major claims that Van Fraassen made in Scientific Image. Well, major claims. The first of them is a characterization of what he took to be scientific realism. But it was a very persuasive one, and everybody more or less went along with it. So science aims to give us, in its theories, a literally true story of what the world is like, and acceptance of a scientific theory involves the belief, involves the belief that it is true. So there's been some distortion of the image, I see. Constructive empiricism. Science, which is Van Fraassen's alternative, new, novel, bold form of empiricism. Science aims to give us theories which are empirically adequate. And acceptance of a theory involves as belief only that it is empirically adequate. So what is this empirical adequacy? It is adequate to unmediated perception. Not through microscopes, not through particle accelerated detectors, no, with the naked eye. So what is empirically adequate is what is adequate to what can be seen with the naked eye. So yeah, this was, I suppose, a nod to empiricism. But he was just, as I say, uninterested in really how you go from what is incident on the eye to ordinary objects. Right, so now here, let me come to the one place where they did, in a way, there's another corruption in the text that's a bit unfortunate, and then I'll try to read my way through it. So there was one point in the Scientific Image where Van Fraassen did address Quinean concerns, and it was here. And it was an objection to Van Fraassen. He was saying, well, what about this objection, what about that? And he's entertaining one. And I should perhaps elaborate a little more on Van Fraassen's position. His position is, if empirical adequacy is all that you need, and that just means whatever is adequate to perception, and that's the purpose of science, right? So, and all that is involved in acceptance of a theory is empirical adequacy. I just do want to put it to you, is this a good way to try to get funding for a new $20 billion high-energy particle physics accelerator? I mean, really, think about it. So the point of the accelerator is to create a huge amount of additional data, which is then the point of science to correctly get right on. And that's the point of the enterprise. So it's problematic from the point of view of driving hearts and minds somehow. And that was always glaringly obvious. But nevertheless, it was a rigorous and clearly thought out, beautifully constructed and well-designed system of thought in this constructive empiricism, which did not fall prey to so many of the pitfalls that ordinary realism, scientific realism, fell to. For example, truth. Well, what if it isn't exactly true? Is approximate truth? Oh, well, what is approximate truth exactly? And you can also ask this. How many scientists actually do believe their theories that they accept to be true? Do they? Well, and you can talk of degree of acceptance and so on and so forth. But it was a very systematic setup, proposition. But now here's the pushback. So Van Fraassen is saying we don't have to believe in the unobservable entities. That's not a part of what acceptance of a theory is. It's not a part of what science is trying to do. It's only trying to get right on the appearances. So, but then the pushback, well, then surely the observable objects and processes we recognize in our world, and here I'm reading from the screen, are also postulated entities, believed in because they best explain and systematize the sense experiences or series of sense data which are at, well anyway, that are all that are available to us. Okay, and Van Frausen's reply is, I mention this objection only because it astonishes me, since philosophers spent the first 5 decades of the century refuting the presuppositions that lie behind it. He goes on, I wish merely to be agnostic about the existence of the unobservable aspects of science. He's an anti-realist, he doesn't believe in them, he doesn't have to believe in them, but he can be agnostic about them. But sense data, I am quite sure, do not exist. So, I mean, this is a very direct opposition between very significant philosophical movements, and it's a shame it was never, in my view anyway, having lived through this period and known both of these people quite well, a little short of tragic. Because I do want to go back to what Van Frausen said was, let me just have a glass of water. I think I better. How do I go back actually? Sense data. Refuting the presuppositions that lay behind it. Sorry, very sorry.
Some people want to hear you by microphone.
Am I not speaking loudly enough? I do apologize. So the presuppositions Van Frausen speaks of. So sense data is the issue. So it was very, very common throughout the 17th, 18th, into the 19th century that we all, what we see and hear and touch and smell and feel are our own sense data. Okay, and this was still dominated in the first 20, 30 years of the 20th century. And logical positivism was somewhat steeped in it. But not really, because there was Wittgenstein after all. You know, there were other very major figures in philosophy who were driving forward a new conception, reacting against sense data. There were no such things as sense data. That entire project of starting with the sense data is radically misconceived. And that became an absolute dominant view, and Van Frausen correctly reports it as such, through the first 5 decades of this century, pretty well. So he asked, what are the presuppositions? And, I mean, sure, you can think, oh, the presuppositions were what those guys were articulating 100 years ago, perhaps. But go back 200 years, go back 300 years, go back to where it all started, which is the drawing I have before you, and Descartes. I mean, the problem, as it were, that science sets to philosophy in the 17th century, and I think it was for the very first time, was of evidence, as it were, just conclusive evidence that our whole perceptual faculty, when it comes to sight at least, is indirect. It has to be. It is the entire physics, the optics of the way the eye works, the eyeball, the lens, the projection onto the back of the eye, and so on and so forth, became well understood. And by Descartes, one of the leading figure, who, after all, was got at the law of refraction, roughly the same time, I think, as Snell. And here is his drawing. And it was a thorough comprehension at this time, in the early 17th century, that indeed, the visual perception at least is indirect. So forgive me for asking, but I'm going to ask you to do something rather special. So touch an eye with your finger, one finger, just gently on your eyelid, and then just push it, look through that, close the other eye, look through the eye, and then just push gently, just push gently. Yeah, right, so I think I've just given you absolutely conclusive evidence that what you see is entirely bound up with what is happening in your eyeball. End of story. I mean, this is the extraordinary and shocking truth that science, as it were, showed to us in the 17th century. And it affected every major philosopher of the time, I think really bar none. And Descartes perhaps was one of the most articulate. Now look at the, it's lovely, isn't it? At the back of the brain, you see, he's projecting the image in the back of the brain. And one wonders, well then, what happens when it's projected at the back of the brain? You know, so there's got to be something else that somehow perceives the image, and so on and so forth. And of course, Descartes didn't know how to deal with this situation, but he appreciated there seems to be a fundamental disconnect between mind and body, and his two types of substances, which rather dominated modern philosophy for the next 400 years, really, one way or another. Although, of course, there are many other philosophers that played a more dominant theme thereafter, but Descartes is still somehow relevant. So I think we do remain in the situation. I'm moving. Yeah, I think we must deal, and the point is only how do you deal with it, it's a fact. So we deal, all sensory input is stimuluses to our sensory surfaces. That's it. On the basis of that, we acquire a grasp and a competence in dealing with everyday objects. And much more than that, well, significant, maybe not much more than that, maybe somewhat more peripheral than we think, an ability and competence in dealing with language and verbal exchange and so forth. And of course, Quine was very preoccupied also with this question of how do these verbal exchanges go, how are they acquired, how does the notion of objecthood first arise in language. And also he thought in neuroscience, but Quine didn't know neuroscience, this was for neuroscientists to sort of figure out, but there was clearly a project here that was essential to our grasp of objecthood and with it of existence, of reality, of ourselves within a reality and so forth. Okay. So all of that is being abrogated really by Van Fraassen. It's just being injected as of a significant epistemological import, at least when you're doing philosophy of science. Okay, but now look at the parallels. So I read again. Here is what Van Fraassen said as his imagined opponent coming back at him. And he was going to say, no, but this is nonsense, you know, only surprising me you're still bringing up that sort of thing. Well, what happens if this is also correct and unobservable objects and processes are postulated entities? So the difference was before with ordinary things being postulated, now it's unobservable things being postulated. And surely in both cases it's the same. We are postulating in both cases the same. Believed because they best explain and systematize a noun sense of sensory stimulus, it's experimental data, or series of experimental data, which are at bottom the only real, and again, I keep losing the text, which are at bottom the only real evidence that we have. I think the wording is identical, the only real evidence we have. So you see the correspondence. You know, on the one hand, we project a world of ordinary things on the basis of sensory stimulus. And it's tremendously important and profoundly part of what it is to live and be a competent, moving, active being. On the other hand, we project from experimental data to unobservable things, which we also manipulate and deal with and arrange and configure. So there's causal stuff going on in both directions, to ordinary things, just to pick up the glass, but in terms of experimental control is very, it's very much action. So if these two things are alike, I mean, if they are broadly alike, and of course maybe they're not, when you really start to dig down and you find that they're not at all alike. But if anything like this is correct, then I think the overwhelming, I think, implication is that science is, the pursuit of science is exactly the pursuit in our ordinary interactions with things. It's the ability to explore, to find out, to meet, to encounter, to act with grace, with judgment, with caution, with appropriate risk. That entire field of interaction is exactly what's going on in science. But of course with science, it's mostly exploration. and then it becomes control, technology. So it starts off exploration, it goes into control. And so if this is the goal of science, then of course one should have a very different conclusion from what I had before. I don't know if I've got much time. I just did want to add, just for 5 minutes, a bit more of an explanation. Do I have it?
If you have 3 minutes.
3 minutes, can I? Okay, thank you. So, of course, let's go into just the ways in which there are disanalogies here, and they are important. And it isn't the case that Van Fraassen has a silly position. He's got a very important position, actually. And the reason is this. Again, I go back to Quine, and this is in a short piece he wrote. It's 5 pages long. It's called Posits and Reality. It sets out his entire philosophy. So I do recommend it for those who want to quickly understand Quine and where he's coming from. But Quine breaks down 3 kinds of entities in reality, if you like. Sense data. And he's prepared to grant that at this point, because he's writing now in the 50s, he's prepared to grant at this point maybe sense data are kind of like posits. But they're sort of sensory stimuli. They're kind of units of sensory stimuli, something, you know. So, and his sense data are evidentially fundamental. Everyone is beholden to his senses for every hint of bodies. That's just my point. The surfaces of our skin, that's it. Particles, physical particles, he means particle physics, are naturally fundamental. In this kind of way, laws of behavior of these particles afford, so far as we know, the simplest formulation of a general account of what there is. Something like that. Words are blotted out. Okay, so the sort of particle physics, and physics is finding out what's really fundamental. At some level, that must be right. Okay, but then last and not least, ordinary objects are conceptually fundamental. It is by reference to them that the very notions of reality and evidence are acquired. So it's really important. It's only by reference to things, ordinary objects, that we can communicate at all. I can articulate anything. There is no language for our sense data. If we have sense data, we don't know how to talk about them. We don't have an autonomous way of discourse. The only way to have a discourse is by common language with observable things. You've got to be able to relate the spoken word with the thing. Otherwise, you're not going to begin on this project. So we can't just talk about our sense data, and therefore we can't talk about our sense data being explained by objects, right? To talk about them both, you've got to talk about that, and you've got to talk about the sense. If we can't talk about the sense data, the only way to talk about the sense data is in terms of the objects. It's like I'm feeling like I normally feel when I'm lying on the beach and the sun is quite warm and somebody brings me a nice cool break, I don't know what, you know. So you've got to bring a load of objects together and then you can kind of pin down a feeling, right? Okay, so so the point is that but if we could talk about our feelings in these raw ways and our sensory data in an autonomous way, then we would have all kinds of theorizing going on, among them theorizing about whether there exists an external world at all. Right? I mean, if we can just do it all at the level of our sense data, then who cares about the external world? So the thing is that what happens with science, though, and Van Fraassen is right, the data in science are in the public domain. We do have an autonomous discourse for them. We can talk about those data. And so it does become a debatable thing that you can say, well, you know, need there be any, do we have to care about the unobservables? Are we committed to the unobservables? It's just replicating that discourse. But now, in contrast to the sense data and ordinary object thing, in contrast, there is an autonomous language of talking about the data. We can talk about the hard drives at CERN. I mean, we can do it, and we can talk about documents that we send to each other as files, and so forth. So I've exhausted my time. I do apologize. At this point, I ought to go back and offer a third option: not scientific realism, not constructive empiricism, and here would be the third one. It would be something like science as perception. But in light of the last talks, I want to rename it now, from now on, science as imagination.
Okay, thank you very much, Simon. Unfortunately, we had a little bit of trouble hearing all of that, so the questions from this end might not be as many as you'll get from Oxford. So please, everybody speak into microphones nice and clearly. Barnaby, do you want to start with a question there?
Yeah, sure. Do we have any questions for any of the three speakers? Francis.
Hi, thank you for the talk. So could you perhaps elaborate on the third
Hold
option?
on, can we speak up?
Yeah, sure. Can you hear me well?
Yes.
Yeah. Thank you for your talk. And could you elaborate on the third option of science as imagination?
So science is imagination. So the point is that if science primary goal of science is exploration, is perception, is the same sort of goals that confront the baby infant in exploring her environment and learning mastery of things, perceptions, how to coordinate, and also how to attach words and verbalization. So if that entire process is just being replicated at the level of experimental data, and how we change, control, understand non-verbally, the objects of scientific discovery, observation, these very indirectly observed, so what Van Fraassen calls unobservables. So this entire realm of the unobservable, which pretty well kicks in anything below microns, ceases to be observable with the naked eye. So everything from there down, all the way down to 10 to the minus 30 length scales in meters, 10 to the minus, increasingly extraordinary brief intervals of time, 10 to the minus 20 seconds, 10 to the minus 30 seconds, this extraordinary precision of what? What do we call it? Well, we can call it perception. And I was thinking science is perception. But I was caught up with the previous speaker, Andy. It was just, I did find the talk enormously. It seems to me there are many things that I wish to learn from you. But it is the idea that the perception that is not seen, you have to imagine. You can't just look at the scene. You have to somehow go beyond. And that going beyond is all about the perception of the unobservable. So, and imagination is probably the right word for it. And I felt the same with the Einstein quote. Of course, I know that quote, and I love it dearly. So, I think that this may be a better way of trying to convey what is involved in scientific understanding. And it's just very unfortunate that the entire project is so hijacked by a tradition of studying theories laid down in books. And a theory laid down in books, unless it's mathematical and sets you puzzles and problems, it's not the same thing. You're not well, anyway, I mean, I think there are so many different ways of taking this discussion, I don't know where to begin, so I'll shut up.
Do we have a question here? Okay, we'll go back to Oxford. Barnaby?
Yeah, we have a question.
Hi, I have actually two questions, hopefully short ones, which are mainly for Andy, though if other people want to chime in, that's great. The first question is whether you think that the important thing in scientific learning and imagination involves mainly observational experience, or whether, as the classical Piagetians would have suggested, it involves more active experimentation, or both, of course.
Sorry, I wasn't quite sure that was it. Yeah, I mean, I really liked Simon's way of framing this. Actually, it's the point is that observation, the kind of sensory stuff, is crucial, but we can't observe so much of what we need to understand when we're trying to grasp what's actually happening in some kind of causal process. So what we do is we imagine what might be going on, and on the basis of that make predictions about what that should lead to in terms of what is observable. And that's where the kind of coordination between the empirical, the observable, and the imaginative comes into play. I think I don't think that you can do science properly without both of those things. That's why I was placing so much emphasis on careful observation and particularly temporal awareness, because without the kind of attention to the detail of what's going on, and that's a kind of empirical judgment ultimately. But for children, this is just simply about learning to pay attention to the right kinds of things and not let the kind of story about what's happening actually become dominant over what we can actually see and what that tells us about our ideas about what's happening. Does that kind of address your question now?
Yeah, thanks very much. The other question I had was whether you think that there are any differences in the processes involved in learning to understand the physical world versus the biological world.
I think fundamentally not, but they're obviously of different order in many different ways. I mean, it has to be said that that much of my own work has been focused really pretty heavily on physics and the kind of the physical world, but that's really a function of the fact that it's easier to manipulate things, and the time spans that we're typically dealing with for physical events are so much shorter, so you can actually encompass them within a classroom lesson, but also within an experimental situation. We have done some work on biology and biological systems, and as far as we can track that, it looks like pretty much the same kinds of things are happening in terms of understanding of biology. It's just that the time spans and often the degree of complexity that's involved in trying to work out what's actually going on is so much greater than it is in physical systems. I'm sure there were physicists who will be throwing their arms up in horror at the notion that physics might be less complicated than biology, but I kind of think it is. So.
Thanks ever so. Yeah, very interesting. Thanks.
Do we have any questions yet here? Barnaby, do we have any over there?
Yeah, we do. Just one second. Here we go.
Yes. Great. Can you hear me? Yes. I have a question for the second speaker again. And what I'm interested in is kind of, I think, the transition from this non-verbal understanding to a kind of better kind of verbal understanding, because you mentioned these practical examples where you talked about vaccines and climate change, and it seems, at least to me, that once you kind of get into that kind of scientific citizenship, you need to be able to evaluate verbal arguments and maybe to give verbal arguments yourself, right? And it seems that, at least in the beginning, if you have this kind of non-verbal understanding, it's something like, you mentioned the example with the falling objects. It's just about what feels right and what doesn't feel right. And it seems that at some point for scientific citizenship, you need to get from just something feeling right to then being able to give an argument that other people can evaluate and either convince by or not be convinced by. And I was wondering whether you have any research or any ideas about how that might happen?
I totally agree with you. The science at the end of the day is a community enterprise, and if you can't talk about it and share ideas in using verbal understanding, then even for scientists that's a requirement, and certainly that's true for any kind of public debate. I think the point that I was trying to make is that that what seems to be mistaken in terms of initial approaches to teaching children about science is the emphasis on the verbal at that stage, because the consequence of that is frequently that all that they're acquiring is the words about things and not necessarily any kind of grasp of what is actually going on. So my point is basically that you have to start with that kind of imaginative dimension and engagement with the real processes, and then you can start to talk about things, preferably using more precise terminology, language to do so as well, because that's the other kind of dimension is, at the end of the day, any kind of scientific dialogue has to be has to involve a degree of precision, and everyday language isn't really very kind of suitable for doing that. But language is fundamental, yes, absolutely.
Could I just jump in?
Can I ask a question? Don't go away, Andrew. I'd like to try and get a little bit of an interaction between you and Simon.
Well, Simon just wanted to come in on exactly this point, so that's good.
Good. Simon.
Yeah, I mean, well, there's so many points, but just on this, I speak with a physicist is what I know, and they're often not very good at putting things into words, especially when it comes to quantum mechanics, which is mostly what I work in. So there's a great deal of talk of equations and words that really have technical meanings in terms of equations. And to try to articulate in more general ways what's going on in quantum mechanics remains, of course, to this day, one of the great challenges, not just to doing popular science well, but to even knowing how to correctly, as it were, understand or explain quantum mechanics, which remains shrouded in enigma, although I have my own preferred view of that. But it's just a peculiarity, perhaps, about physics. I can well believe, Andy, that in most sciences, the equations are just not so important elsewhere, and the word becomes all important. I'm sure that must be true, or very important anyway.
No, I mean, it's... I guess the way I think about it is that the mathematics and the equations are simply a kind of form of technical expression that works in that particular context. The difficulty with that is sharing that with people who don't have that understanding. But I see, I tend to follow scientific journalism fairly closely, and I actually... it seems to me that there are a number of people now starting to do a pretty good job of trying to explain ideas from quantum physics in a more kind of comprehensible language for a more general public. So I think it's not that it's impossible, it's just that it's perhaps something that people haven't turned their attention to so very much until comparatively recently. But as the public in general becomes more aware of the notions of quantum physics, then I think that's something that is more attractive to try and do, and is certainly something that people are more receptive to. So it's possible. It's just, perhaps a more difficult enterprise in many respects.
I look forward to further discussion.
Thank you.
I'm a many welders myself, Everett interpretation and all that.
Actually, can I push you to discuss a little bit more the role of the imagination? Because I just wondered, Simon, whether Andy is talking about the imagination getting us onto mechanisms. And I'm trying to get clear on what Van Frossen would say to that. Okay? I wonder if you could just...
I think it's perhaps for me, sorry, I was thinking you were addressing Andy. It is perhaps for me to respond. So I think BAS would be fine with everything at the pragmatic level. By all means, use whatever techniques you want, imaginative techniques, absolutely, trances, long hot baths, or he would climb mountains. So there's all sorts of things where he would be perfectly, enthusiastic about people doing these things. But for him, it wouldn't change the essential epistemological situation, which is the evidence is data that we see with our naked eyes. And it's only getting that right that can possibly count as anything to do with science. I mean, it's a very, and do you see, that has to be addressed in a much more full-fronted way, I think, which is what I tried quickly to do. That it is to make use of the fact that we can speak of data in an autonomous way without commitment to unseen entities, that permits us to draw that line in the sand. But if actually all you're doing is you're doing what at the level of sensory stimulus would be kind of insane, right? If that is the parallel, then this surely is a mistake. It's a mistaken idea of what the scientific endeavor is. You know, you might equally say the endeavor is to get it right as to my sensations that I encounter over the next 30 minutes. That is not what I'm attempting to do. And neither is the physicist attempting to get the data right. The physicist is attempting to learn something new. So and imagination, it seems to me, is bound up with that. But you've got to sort of buy into this kind of story before someone like Van Fraassen would be moved by, you know, what about beauty, what about imagination, what about boldness, what about daring, what about originality? I mean, he'll applaud all of those things, but it's just irrelevant to what he sees as the task of science, what the goal of science is. And it is a very passive position to say that what is involved is the theories and acceptance of theory, as though that's the end of it. You know, the theory is written down in a book and there you're done. I mean, that just seems a mistaken idea too. So anyway, I could go
on. I think we're kind of on the same page. I mean, I think, you know, two things. Firstly, as I hope I've kind of tried to make clear, it's not about imagination on its own. It's about imagination provoking an understanding that you can then take back to test against the world. And that seems to me to be absolutely fundamental to how science proceeds. But the other thing, absolutely, is that parcel of that, I think, is the notion that, as I have been telling my students for I don't know how many years, science is provisional. It has to be provisional. It's always about, you know, what we kind of can say at this point in time, what we don't know, and therefore where we can go next. It's always about you know, this is where we are now. It's not where we're going to be in 10 years' time or even next week. It's so it's always about being provisional. I think that's kind of, I get the sense that's what you're saying as well.
Yeah, well, as Popper said, science is built on big, great, huge logs of wood that drive deep into the mud.
Yeah, absolutely.
And may or may not stay put.
Okay, are you having a burning question over there, or shall we draw it to a close, Barnaby?
I think we can finish. Thank you.
Excellent. Thank you so much. I'll say good night from Hamburg. And good night, Oxford. And thank all our speakers. Let's all do that on both sides. I know you're going to have a dinner with some of the students and speaker over there, Barnaby. I hope you have a nice evening, and everybody, good night.
Thank you very much.